Well, I think it was mentioned yesterday by Mike Coates that the batoids, the skates, the rays, the sawfishes and so on tend to be the sort of almost the neglected group of fishes or fish-like forms alive today. But actually, they're a diverse group and they do some very interesting things. I suppose the one thing about them is that in many respects, they appear at least to be quite conservative. And it's easy to come up with a sort of classic separation of different batoid groups in that you have the guitarfishes, these vaguely shark-like things with a long, powerful muscular tail, the sawfish, which are rather similar, but with a big rostrum, the electric rays, strange, modified, flabby things, which generate a powerful electric current, and then the skates and the stingrays, two flat groups that use their pectoral fins as their main propulsion and have a very reduced tail. Recent studies have shown that these groups aren't monophyletic. So what you actually have is these so-called guitarfishes, these things with a rather unmodified body plan and a powerful tail, are present in a whole range of different positions within the tree of relationships of the batoids. And within this, the other groups fit as close sister groups of guitarfishes. So the electric rays in here fit in with the fan rays. The sawfish in there fit in with some of the rhinobatids. The stingrays fit in as really just sort of a derived group within it. The skates sort of fit in a rather sort of nebulous position down near the base of the living batoids. And all of these form a sister group to all of the living sharks, and these in turn form a sister group to the living holocephalans. Now, one interesting thing with this is where the position of this split between the modern sharks and batoids occurs, and what it means as far as our perception of the origin of the gnathostomes, in that this as a clay, as the modern elasmobranchs, the modern chondrichthyans as a clade, are obviously the sister group, and some would say rather poorly studied and of low interest sister group to all of the other gnathostomes. So if we can find features that are common within these and basal gnathostomes, we can be fairly sure that we have a sort of a basal character, as opposed to just by looking at one of the big gnathostome branches versus the other, we are going to be a little bit more biased because we don't know what is a derived or a primitive state. As for the split between the modern living sharks and the living batoids, well, it certainly has occurred by the early Jurassic. So this is a nice, beautifully preserved guitarfish-like form from the late Jurassic, but it does have some features that seem to place it outside the clade of all the living forms, although it just looks like yet another of these, this great morass of guitarfishes. Among other things, it has fin spines, which no living batoid does. We also have typically batoid teeth, which are very common from the late early Jurassic onwards. These, by the way, are from the Bathonian, from the Middle Jurassic, of a place called Wood-Eaton Quarry in central England, of which there's quite a nice poster downstairs. It's arguable as to whether some of these strange teeth of unknown affinities that turn up in the Upper Triassic can be assigned to the batoids or not. And to be honest, I'll leave that to people who know more about the Triassic weird stuff than I do. Just to sort of, so what I want to do is to look at the development of the dentitions of some modern batoids and have a think about what that actually means and what it might mean in respect to other gnathostomes. Well, this is a scan of an external view of the jaw of a fairly typical batoid. This is a female of the thornback ray, Raja clavata, which is probably the commonest European batoid. Here we've sort of sliced back through from back to front, so you can see the underside of the teeth, and you can see the patterning. So they are in vertical files and horizontal rows, and the teeth actually alternate. So we have this wonderful alternate pattern. You can also see that each of the teeth have two distinct root lobes. This is a cross-section at right angles through this lower jaw, and you can see how the teeth form. The teeth are formed continuously in the dental lamina down at the back of the jaw, progress upwards through the oral side of the jaw until they're fully developed, move over into the occlusal surface where the teeth do their work. They wear down very rapidly, move out of their functional position, and then they drop out. So we've got a continuous replacement of teeth. There are other features that batoid teeth show that are quite interesting and give a bit more variation into them. So this is the mid part of the upper jaw of Raja clavata again. This is a female. You can see the teeth are all rhombic, flat. They look pretty much the same. This is a male of the same species. Teeth look utterly different. And there appears to be evidence that the shape of the teeth of this species at least changes through the year. These don't appear to have a different diet. It just appears that as an animal with internal fertilization and no hands, they have to hang on somehow. We've got other features we can see in batoid dentitions too. This is the shark ray, a thing called rhina, and you can see here, again, these teeth closely packed, and the jaw is folded into a set of crenulations, which increase the sort of crushing power of the jaws. We've also got some strange deformation up there where some of the files of teeth haven't developed properly. These are the jaws of a small stingray, a thing called neotrigon, which is mostly these small little rhombic teeth, typical of many batoids, but just at the curvature of the jaw, we have some big pointed, what I'm almost calling pseudo-canines. And in some batoids, the teeth are very highly derived. So majority have these very small, rather rhombic teeth, but in some, they're very modified. This is the lower jaw of the eagle ray, Etabatus. In this case, just the central row of teeth, the symphysial row, are present. All the other teeth have disappeared. So we just have these huge chevron-shaped symphysial teeth, again, forming at the back and then moving forwards. You can also see here, there's your grinding surface. That's where they work. This is all polished out here. That's post-occlusal and appears to be actually used as a spade to help it dig up invertebrate food. All right, so what do we know about chondrichthyan dentitions? Well, most work has been done on the sharks because they're more media-friendly, more, we could say, grant body-friendly, I suppose. So this is your classic workshop of the sharks. This is the spotted dogfish, Scaliarinus, and these are the teeth of it. Like with the batoids, you can see the teeth in this alternate, although in some sharks, the teeth don't alternate and are present in single files. But in many ways, these are fairly similar. But we do know from the development of this and the gene expression for the teeth in this is that as the teeth develop in the embryo of Scaliarinus, they develop one row at a time, with each row starting with the development of the symphysial tooth, the tooth on the border, on the symphysis of the two jaws, and sort of the mineralization and development sweeping backwards, row at a time. So from front to back, front to back, front to back. And with the first formed row having the symphysial tooth, even though in some sharks that's secondarily lost during development. When we look at the batoids, we can see, in some cases, similar patterns. So this is a guitarfish. This is a thing called rhinobatos, the classic of the group. And this is the jaw of an embryo, which still had a fairly substantial yolk sac attached. That jaw is, well, way less than 10 millimeters across. And you can here just about see the first formed teeth. So they're alternate. There's a symphysial tooth there, and they're spread right along the length of the jaw. So just like in Scaliorhinus, the first row of teeth of Rhinobatos form right the whole length of the jaw and include a symphysial tooth. So although we have yet to test it, it seems likely that likewise the germination of these teeth sort of sweeps backwards from the symphysis to the commissure. Within other batoids, the pattern appears somewhat different. This is a rather underived electric ray. There's a thing called discopygy. The more derived electric rays are rather difficult things to work with because they've changed their body form so much to generate powerful electricity. It means that most of their food is dead or stunned by the time they eat it. So the teeth don't actually do very much. This thing uses electricity more to get around, and so it has sort of normal teeth that do a normal toothy job. You can see here in the lower jaw of an adult, you can see the alternating tooth rows. And there's quite a lot of teeth in there. So that's the symphysial tooth. So we've got a lot of teeth there, including the symphysial. The next row behind doesn't have a symphysial, but again has quite a number of teeth. If, however, we look at some rendered CT images of an embryo of this, or a late-stage embryo, we find something slightly different. So at the bottom here, this is the outer view of the jaws of an embryo discopygy. So this is the upper jaw and the lower. And what you can see is there's the first formed teeth. There is no symphysial tooth, but we have two teeth, one either side of the symphysis. The same on the lower jaw, two teeth. The next row has 3, 2, 3, including a symphysial. If we take that upper jaw and roll it a little bit, so we can look sort of from that direction, there's the first 2 teeth, there's the next 3, and we can just see 4 in the following row behind that. So we rotate it a bit more. There's the first 2 teeth, 1, 2, 3 of the second row, 2, 3, 4 of the third, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6, and so on. So each successive tooth row is actually adding an extra tooth. And the first tooth row doesn't contain a symphysial. So that seems a very, very different pattern. So I thought, okay, is this a structure limited to some stingrays? We then had a look at these other highly derived rays, the myliobatids. So there we have an adult myliobatis. That's the lower dentition of a neonate, very small, and you can see these enlarged symphysial teeth, but on either side you have the teeth of 1, 2, 3, 4 without a symphysial, and 1, 2, 3 including one. Slice it vertically, you can see these teeth are highly modified, they lock together, and so on. If we look at an embryo of this, this is a rendered surface view of an embryo, upper jaw and lower. First row of teeth, 1, 2 teeth, no symphysial. Next one, 3, including a symphysial. On the lower jaw, the same pattern. Here, looking at the sort of outer teeth of a neonate, which isn't much bigger than this embryo, the teeth have already established this alternating 3, 4 pattern, and the teeth are already locking together. So we have an increase in tooth number for just the first 2 rows, and then it stops. And then we retain that same tooth number for the rest of the ontogeny. At the same time, we can see the number of root lobes improve. This is a very difficult image to render, but here in the embryo, you can see the teeth have 2 nutritive grooves, or 1 in lateral teeth. The neonate, we're getting more. In the adult, we've got shed loads. So, whereas in the rhinobatos, we have this development pattern which is the same as in Scaliorhinus, with a lot in the first row, including a symphysial. With the Discopygi, we only have 2 teeth in the first row, and they add subsequently. In Myliabatis, the same, up to a maximum number. And in that Etabatus, with only symphysial teeth, the first row actually contains 2 little tiny teeth. So when we look at how these fit in phylogenetically, it appears that we have a basic pattern in Rhinobatos and Scaliorhinus, and these others with a progressive addition of teeth are found in rather more derived groups. So hopefully that's given you a bit of a taste that rays are rather more interesting.
Thank you very much.